As aquaculture attempts to move away from becoming dependent on fish meal and oils, farmers must ensure that feed continues to provide the best nutritional profile for the well-being of their animals. This includes supplementing vitamins and minerals that would otherwise be provided through fish meal and fish oil. Research is focused on microalgae as primary producers for omega-3 fatty acids, potentially replacing expensive and unsustainable fish oil from fisheries. Until now, little research has been done on its additional potential for mineral supplementation.
Here at Wageningen, what we are doing is we're actually putting these minerals into microalgae first, so that they can take it up in an organic form, and then we can supply these microalgae within the aquafeed. Firstly, we've looked also into how can we better assess minerals in aquaculture and in microalgae, how to prepare the samples for mineral analysis. And then we've scaled it up, looked at the toxicity, bioaccumulation, and how can this work in a large-scale facility, such as Algae Park, our research facility where we can do that here at Wageningen. And what we have done is we've actually incorporated selenium into the microalgae, and then we've tested to see if this incorporation means that when it's provided to fish, they will take it up in a more efficient way and create less waste in the aquaculture stream.
Selenium is essential for fish development and immune system functioning. Deficiencies can affect growth, increase mortalities, and hinder the immune response. But not all supplements are created equal. Some inorganic mineral mixes are less bioavailable.
Minerals are still a challenge in aquafeed, since many of them are inorganic and they're not readily absorbed by fish.
Researchers have found that selenium-enriched microalgae is a more bioavailable source of the mineral in comparison to traditional inorganic mineral mixes containing selenium. Selenium in microalgae can be stored in amino acids, which are fundamental in the fish diet.
The microalgae that we have produced at Algae Park has been sent to Portugal to a company called Spados that is specialized in aquafeed. This has already been administered to fish as a selenium-enriched ingredient, and we are now looking at the preliminary results of this study, where sea bream has actually taken up selenium-enriched microalgae. So for us, that is a success.
The selenium content of terrestrial-sourced ingredients varies based on the soil composition and lacks the optimum ratios of amino acids. In contrast, the uptake of selenium into the microalgae can be controlled, so that the selenium content can be specified to the needs of each fish and each developmental stage of production. In this way, selenium-enriched algae is not only a more efficient use of resources, but also improves the welfare and associated productivity of the animals.
We have an example of some of the algae that we have studied, which is a strain called Nanocropsis oceanica. It's a marine strain that is already used in aquafeed.
While fish can digest a variety of microalgae species, not all microalgae species can be enriched with the required quantities of selenium needed for aquafeeds. This marine microalgae species was selected for its robustness, its high EPA content, and its ability to grow in a variety of cultivation media.
Our first work within our project looked into how to wash biomass cells, in this case our microalgae cells, so that we would have a more accurate determination of the mineral content. Since this is very important because this is what we're going to give to the fish as a final product.
The biomass harvested was dried and sent to the industrial partner Sparos, who incorporated the microalgae into the aquafeed for further fish trials within the project.
We provided over 12 kilograms of biomass to the company that is working with us called Spados.
Future work will aim to evaluate the bioavailability of selenium in other ingredients, such as selenium-enriched yeast and macroalgae with fish feed trials in sea bream. Make sure to check out the publication in the link below if you're interested in learning more about the work.